So my name is Hiroki Ueda from Japan. I have a little bit Zoom jet lag because I'm a night person. And then this is 6 a.m. in the morning. Okay, so today we're gonna have some kind of a discussion, maybe severe discussion on the single sample, sampling. And then also we want to talk about what can we know about the, our body time by using just single time point sampling. Okay. So today we have excellent speakers, 5 panelists from all over the world. And then actually Takeshi is also from Japan. He's in the same time zone of me. But Achim, I'm sorry for Achim, because he is from Berlin, and then he is now in the 11 p.m. And also we have Derk Jan Dijk from Surrey. He is in the 10 p.m. Okay. And then we also have Jake and Rosemary from Nashville and Chicago, which indicate that we are living under clockless and sleepless society, okay? And then, therefore, we have some, we feel that some importance to know about our time, right? And then during this debate, I like to have audience put your question onto the Q&A kind of a chat. And then Catherine will pick up your question in the end of this debate and then give the microphone to you. So in that, moment, please ask the question directly to the panelist or moderator. Okay. All right. So, do you agree? We have we feel the importance to know your time. Okay. But you know, our time is not always is social time, right? And also our time information is important and valuable in terms of some medical applications. Because, you know, this is a kind of a famous example done by Francis Levy. And then he nicely showed the chronotherapy is, is possible for colon cancer treatment. And also, okay, recently, John Hargness and his colleagues indicate the target of our FDA-approved medicine is oscillating. So therefore, you know, dosing time is very, very important for your health, right? But there is a problem. So because, you know, body time detection is a little bit hard. So here is a pioneering work by Levy and Sack. And then they showed the plasma dim light melatonin onset can be used as a marker of our circadian time. And then this little bit invasive method can be non-invasive because, you know, which is done by Kenaway and also his colleagues. Because, you know, soluble DMO is also possible to show your circadian time. But, you know, this classical method is a little bit, stressful because, you know, you have to be sampled every 2 hours over at least 1 day to show your circadian time, okay? So therefore, you know, there is some kind of solutions given by botanist Linne. And then his idea is to use the flower clocks, you know? So in this flower clock concept, there are many flowers, okay? So opening at the different time. For example, some flower open from 6 a.m. to noon. The other flower open probably, how to say, other time zones. And then by watching these flowers, you can tell your time, okay? So here's a classical method. So, and then which is a long-term observation, right? And then if you look at the flower and then look at the opening and the closing of the flowers and then tell your time. But a new method, single time point based method is just one time observation of many flowers. And then you use some kind of timetable of the flowers and then you, how to say, use the pattern, closing and opening patterns of flower to tell your time, okay? So, and then realization was done as early as, 2004. And then there are, at that time, there are many studies of microarray are published, right? Including, how to say, John Hogness lab and our lab, and also many other labs in the world. So by using microarray data, so there were morning genes and daytime genes and evening genes and late night genes. And then using these information, so at the morning genes are some kind of, opening, okay? And then evening genes are, is closing, okay? And then by using these patterns, you can say, you're at the daytime zones and the evening time zones, and then early night and midnight and late night time zones, okay? This is a kind of a realization, okay? And then first realization was done by using microarray, but not only microarray, but also metabolite can be used to detect your body time, which was done in 2009. And also mass spec or maybe antibody-based detection of the protein may be possible, okay? So many kind of modality can be used to detect your time, okay? And then, of course, application to human clinics is very, very important. So as early as 2010, Akashi lab showed hair follicle, your hair or your beard follicle has a very, how to say, beautiful circadian oscillations. And she nicely shows that is a time indicating genes in the hair follicles. And also in the blood, especially blood metabolomics or maybe transcriptomics show the nice oscillations, okay? So blood metabolomics is done in 2012. And then in this case, double time point sampling, but by using this method, you can tell your time. And then Devla Skinny lab nicely shows blood or urine has a circadian or time indicating substances. And then Dijk, Derk Jan Dijk, and also Jake lab nicely shows whole blood transcriptomics indicate the circadian oscillations. And then Achim Kramer's lab nicely showed blood monocyte have a circadian oscillations. And then John Hogness lab nicely shows the skin or various other organs also have circadian oscillations. Okay. I could not include many others application, but I'm so sorry about this. But if you think your citation is important, please put into the chat or Q&A comments. I like to include many, other important citations. Okay, so we are so much focused on the human because we are human, right? But please don't forget about the other application. For example, animal physiology and plant physiology, because one of the important things in our field is the diversity, right? And then, for example, here's one application, possible application. So by using this kind of a single time point measurement technique, we apply this method to photoperiodisms. And then Takeshi Izawa applied a single time point method to the rice in the field. And then here is an important example of single cell transcriptomics done by Derk Jan Dijk. And then he nicely showed even single cell have a time indicating genes, and you will get the time course or timetable of these time indicating genes. Okay. All right. So you might think of many applications of this method. Okay. And then since 2004, there are many update or breakthrough in this field. I just show you some breakthrough, which include Cyclops method. Okay, so in the original method, we have to have time course sampling, but by using clever idea by John Hognash and his colleague, they tried to use a kind of a randomized data, and then try to identify in unsupervised manner time indicating genes. Okay, and then many and also very sophisticated algorithm apply to this method. For example, Jake nicely developed the Zeiger software or algorithm in 2016. And then collaboration between Sason Kolshi lab and the Baldi lab developed the BioClock algorithms or software, which is based on the deep neural network techniques. And then, and also Derk Jan's lab using the partial least square regression method to detect our time. And also Rosemary lab developed the time signature software, which is quite robust and then applicable to many platforms. Okay, so we have 5 excellent panelists here. And then I just pass my microphone to Derk Jan to give some introductions. Okay, so please go.
Well, thank you, Hiro, and good time to all 327 participants, and of course, including the panelists. I'm not sure, Hiro, whether you can show my slide. Yes, there it comes. I think that, or I hope that this debate will not just be about methods, but actually about what these methods are trying to accomplish. In the title of this session, we use the word body time. But what is it that we actually mean by the word body time? We now know that there are oscillators everywhere in our body. So if we take a sample from the skin and assess time in skin, do we want to know time in skin, or do we want to know time elsewhere in the body? Likewise, if we take a blood sample, what is the target? Do we want to know the time in blood, or are we looking for some kind of a signal that gives us information about time in the SCN. So our approach has been to look for methods that could give us, on the basis of blood samples, information about the timing of the melatonin rhythm, which of course is considered to be a marker of SCN phase. So I think that's one important point for our discussion. What is it that we want to accomplish? The next thing we want to know, is our method really giving us answers under various conditions? For example, if I take a blood sample in the middle of the day, but I have been sleep deprived for 24 hours, or I am sleeping during the day, does this blood sample still give us information about our target, in our case, the SCN? That's why we designed this experiment where we collected quite a few RNA samples under various conditions. Condition number 1, people are sleeping during the night. Condition 2, people are sleeping during the day. Condition 3 and 4, people are awake, and they have been awake for quite a while, but in one condition they were well rested, and in the other condition they were not well rested. And this, I think, is one important aspect. Do we really believe that when we collect samples under different conditions, these markers still represent the phase of our target? And that's of course relevant for applications. If you want to know the phase in a shift worker, the shift worker may be sleeping during the day. You don't want sleeping during the day to have an impact on your marker so that it no longer reflects the phase of, for example, the circadian pacemaker. In the left corner of the slide, you see a distribution of phases of rhythmic transcripts. In the black polar plot, you see the distribution and frequency of rhythmic transcripts when you're sleeping in phase. The red little blob in the middle represents the number of rhythmic transcripts and the distribution of your acrophases when you're sleeping during the day. And you see the massive masking effects on these markers. That's why I think it is important to test our markers under these conditions, in particular under conditions in which the sleep-wake and other behavioral cycles are displaced. So how well our methods actually perform is maybe, in terms of accuracy, is less important. I think it's more important to establish how robust they are. We used 1 sample, 2 samples, 3 samples, 2 sample differential, and got reasonable performance. But importantly, that performance was robust against, for example, the displacement of the sleep cycle. So my message is, we need to be clear about what our target is, and we need to demonstrate that our marker is robust against manipulations so that the target phase is still represented in our marker. And that's all I have to say.
Thank you, Derk Jan. Okay, so, all right. Yeah, so Jake, are you ready?
Yeah.
Yep.
Can you hear me?
Yep.
Sure. Okay. Okay, good. Yeah, so thank you, Hiro. Thanks, Derk Jan. I think that was great. Totally agree with that. So as Hiro mentioned, the method that I've been involved in developing is called Zeitzeiger, which comes from, related to Zeitgeber. And so zeigen is to show. And so, as all the methods you're going to hear about, it's a supervised learning problem, right? So you've measured a bunch of features in many observations. And in this case, the thing we want to predict is something like circadian time, some circular variable. And in the case of Zeitzeiger, it uses what are called sparse principal components to learn how these features change over the course of the day, let's say. And essentially, let's say, it's going to use 2 sparse principal components. That's a parameter that, you're going to see what works best, like Derk Jan was saying, but in the case of 2 SBCs, the goal is to essentially learn a clock that tells military time based on your training data. so that now when you get, oh, can you go to the next part, Hira? The next slide. Yes, thank you. Great. So now when you get a new observation, right, the goal is to try to place that test observation for which you don't know the time onto your military clock, basically, and say, all right, what is the time of day that is closest and how far away is it? And again, you know, you have to make the decision about what are you really trying to predict, right? What is that time? Is it Dilmo? Is it something completely related to in a different organ? And then test, all right, how well does that work on average? So, you know, we published a couple papers on it. It's publicly available. There's documentation. And one of the cool things, so we applied it to mice and humans, all on publicly available data. Here, if you can go to the next part. Thanks. And so what you can see in real, you know, mouse data, right, is so in this plot now on the bottom right, every point is a sample, and we're plotting the SBC1 versus SBC2 for a totally different data set that we didn't train on, where we have wild type and BMAL1 knockout samples from mouse skeletal muscle. And you can see that in wild type, the samples are basically following the pattern we already learned. Whereas in the BMAL1 knockout, the specific to skeletal muscle, there's still an oscillation, but it's actually shifted, right? And you can see that based on the SPCs that Zeitziger learned. And, you know, this was mouse data at the time, and so I was kind of skeptical about whether it was ever going to work in humans, but now it turns out you know, this sort of principle could apply. And I think it's worth thinking about, like, what would this actually look like in humans for different sorts of diseases or different sorts of conditions. And I just want to close with two discussion points, maybe, is that this idea of there's no free lunch, right? No one method is going to work best in all situations. No one parameter set is going to work best in all situations. And then sort of like Derk Jan said, just to emphasize, I think one of the challenges is going to be trying to measure and then potentially influence the right property of the right rhythm in the right tissue for the right phenotype. So do we want to try to measure amplitude of a particular metabolite in the liver for people who have type 2 diabetes, or, you know, fill in the blank of all those things? And so I think we have a long way to go for that.
Thank you, Jake. All right, so next, Achim from Berlin. Are you ready?
Yes, I'm ready.
Yep,
so
go ahead.
Thanks, Hiro, for organizing this exciting session. So we have developed a method which predicts internal phase of entrainment from a single blood sample with extremely high accuracy. Its median deviation from Dilmo, you see that on the right part, is only about half an hour, and this is independent of whether you draw a blood sample in the morning or in the afternoon. So I think this remarkable accuracy is mainly due to two factors. Firstly, we decided to focus on a specific cell type in the blood, the monocytes. We have previously shown that monocytes possess a high amplitude circadian clock, in contrast to B cells and T cells, which are barely rhythmic, but make up about 80% of all white blood cells. Although sorting out monocytes is an extra step, but this really pays off. The second reason is that our platform to measure our biomarker transcripts is NanoString, a highly robust multiplex platform that directly counts molecules and does not require reverse transcription or amplification steps. We performed two studies to develop our test. The first study was a carefully controlled constant routine study with young males to discover the biomarkers. And the second study was completely independent. And I think this is really an important point for any diagnostic assay development. You need a completely independent validation, not just only divide, test, and training samples, because that's the real life sampling is really important. We used here in this second study a real life scenario and included females and extreme chronotypes. And the results of this validation study, you see that on the right. This is about half an hour accuracy. Important to say is that our method is independent on the bioinformatics methods for biomarker discovery. We derived models using Zeitzeige, which just Jake has nicely shown, also lasso or partial least square. They all perform equally well. I think it's really the cell type and the platform which makes the difference. And I just want to say 2 words for Derk Jan. I think this is really an important issue, the robustness. But I think there's a trade-off between robustness and accuracy, because as Jake said, it really depends on the application. You cannot develop a method which is both working in all kinds of scenarios and it's at the same time as accurate as possible. I think this is something for the discussion. And also the question is, do we really need to be super accurate? This is for now from my side.
Thank you, Achim. All right, Takeshi Izawa. So are you ready, Takeshi?
Yeah, I'm ready. Thank you, Hiro. So today I'm telling you on our approach to estimate circadian time crop, especially rice, growing under natural fluctuating environment. So first of all, I'd like to tell you one of the big difference between crop cases and human cases, as the speaker already talked about. That is, for crops, we want to look at the clonal propagation of crop in the field. For example, around 40% paddy field in Japan are occupied with an elite rice cultivar termed koshihikari. So in terms of circadian time estimation, we don't have to care about genetic variation or heterogeneity so much. In other words, my talk is in the kind of physiological analysis. So in addition to this situation, many think that the naturally fluctuating environment results in big noises when we try to estimate circadian time, but I don't think so. I'm thinking about fluctuating environment factor should be considered as a strong determinant for gene expression in plants. Based on this idea, we have first developed a mathematical model to explain each gene expression as a function of environmental factors such as ambient temperature and radiation. We used more than 400 transcriptome data from field sampling and succeeded to develop a model for most circadian clock-related genes. So those models were with more than 0.8 R2 coefficient of determinant for test data. So with this model, we calculated gene expression of around 30 crop-related gene with 10-minute intervals during the entire crop season in a year. Based on these calculated gene expression data, we try to estimate circadian time in a similar way of Jake's Zeitzeige method, although we didn't use sparse principal component analysis. Instead, we just used a simple Bayesian inference. To do that, as you see, we made a heat map between time of day and gene expression most of circadian clocks. Then we can get posterior probability distribution when value of gene expression were given. With this way, when we combine the posterior probability distribution of time of day for distinct gene in an RNA sample, we are able to narrow down the circadian time nicely. Then we searched for best combination for crop genes and found 16 clock gene related genes were good enough to get the best performance prediction for test samples. At the moment, we got around 24 minutes as a mean of absolute error for any time of day. I show you here the case of test data, that is 1 minute interval sequential sampling data from field. So I hope you see the same time progression among 1 minute samples. So this case, the absolute error relatively expectation was around 7 minutes. I think there are several reasons for this accuracy in our systems. One of them is that the plant circadian clock may have an elegant system to integrate environmental factor information to determine the phase of circadian clock. As you see, this is the case of the OS gigantea or OS LHY gene have a distinct gate timing for temperature sensitivity. Finally, here I want to some example for application in my talk. Thus, I here present the data to predict flowering time of rice from the G expression data using a related model. This model was just made simply by loss regression. As you see, we can predict flowering time. This is a very important agronomical trait of rice with a sample at 60 days before sampling, before flowering with a few days errors. So I hope that you have realized that field transcriptome data contain valuable information for future agricultures. So I'll stop here today. Thank you.
Thanks, Takeshi. Okay. So now all panelists give some introductions. So therefore, I'd like to open a, discussion sessions. So we have a couple of discussion topic here. For example, okay. So we have a couple of discussion points here. So first, we have some kind of variety of different method. Some focusing on some kind of detections, some focusing on some kind of analysis, and then many of the panelists talked about the accuracy and also robustness, right? So okay, so I'd like to have the comment from Derk Jan first, and then open some comment to have some other comment from other panelists. So could you start, Derk Jan?
Sure, well, thank you, Hiro. And before I forget, you of course have been a pioneer in this area, both in animals and humans, and you were decades ahead of us. Anyway, but here we are, and I think some of the important questions still need to be answered. I felt that there is some agreement among the panel members that the actual methods, the algorithms, are not necessarily that critical, and that some may be slightly better than others. I think that there is some kind of agreement that the source of the sample may be important, and I can fully accept that whole blood may not be the best method, although it may be the easiest source because you don't need to sell sorting steps. So I think that a main discussion for me remains the robustness. And Achim said, well, there is not going to be one method for all scenarios. I fear that in certain clinical applications, it's unclear which scenario actually applies. If a patient comes to the clinic and has a sleep problem, we don't necessarily know whether this person is desynchronized, SCN versus sleep-wake cycle, or whether this person is synchronized. So that's our approach was, we take samples from all kinds of scenarios and develop that method in that way. And these are all of my comments.
Okay, so do you have any comment, Achim?
So first of all, just to clarify one little point, because there was a misunderstanding about the source. So we use, and that's what Rosemary means, we use monocyte. PBMCs is basically all white blood cells. So I think Derk Jan uses PBMCs, we use monocytes. Whole blood is basically the same as PBMCs, just to clarify that for the audience.
Right. Okay.
And in terms of robustness, I think Derk Jan is completely right. I just want to add some little comments. So first of all, Derk Jan, do you know how robust Dilmo is? Because you train against Dilmo under situations where sleep-wake cycle is disturbed. And if you train an algorithm with something which you consider a gold standard, then you need to know that this gold standard is a gold standard under all kinds of situations.
Yeah, no, I agree with that comment.
Do we really know that?
Let me comment on that. If you look in forced desynchrony experiments, if you do them under dim light and you look at the progression of phase of melatonin and cortisol, that progression is actually the same and clearly desynchronizes from the sleep-wake cycle. So under these conditions of dim light, we believe that melatonin and cortisol are an indicator of something that relates to SCN signals that, for example, drive the sleep-wake propensity rhythm. But there are, of course, for melatonin, important potential confounders like light. You know, shine 20 lux of light or 50 lux of light on an individual, and your melatonin is no longer a robust indicator of SCN phase. You have seen the wonderful study coming out of Monash from Andrew Phillips and others in PNAS showing that in some participants, 10 lux already suppresses melatonin to a considerable extent. So the question of robustness applies also to these gold standards. I agree with that, and will apply, but that's why we need to have this discussion and clarity around this. How the melatonin phase always truly represents SCN phase, we don't really know, because in principle, SCN phase, how do you define it? On the basis of a light phase response curve, and etc. So I agree with your general point, but I still believe that melatonin and cortisol can give you quite interesting and informative information about SCN phase.
Okay, so Jake or Rosemary, so Achim and Derk Jan have, how to say, some kind of experimental experiences, and then Rosemary and Jake, so you're mainly focusing on the analysis part. So do you have a comment from that point? So Rosemary, could you say?
Yeah, happy to comment. So certainly what we've seen is that the differences between the methods are not as strong as the differences between the experimental sources of data. Nevertheless, there are differences between the method's performance. Something that we've seen as we've been trying to address this question of normalization and cross-study performance is that these normalization steps that we're taking in our algorithm can actually be used as inputs to any of the other algorithms. And what we've seen is that doing that pre-processing normalization step helps in all cases. It helps Zeitzeiger's performance, it helps the PLSR method's performance, it helps time signatures performance. And so I think it's worth thinking about whether there are ways to pre-process the data in order to make the circadian signals stand out from the rest of the noise, right? That's kind of the goal of the first stage of time signature, but I think it could be applied in any context.
Jake?
Yeah, I mean, another thing that I would say just relative, you know, as a person using existing data is, you know, I think a lot about the analogy of the drunk person looking for their keys under the streetlights. And, you know, the data that has existed for a long time has been, you know, whole blood, right? Now there's monocytes. And, you know, it's a lot about things we try to predict, right, that we build the ground truth on are the things that we can measure easily. And so, you know, you just always wonder, like, well, what else, like, could we measure? You know, is there something else? Like, what if we don't want the phase of SCN for our particular application, right? You know, what if we care about something related that's being affected by the feeding cycle? Like, what could we conceivably easily measure and collect data on to pass it to, you know, whatever method we want?
Okay. Yeah, so Achim.
So I just wanted to close the accuracy thing. I want to give two thoughts. One is, which I already kind of mentioned, is that it may be worth discussing how accurate do we really need to be? I mean, yeah. I say we are half an hour close to Dilmo, but the real question is, do we need to be half an hour close to Dilmo, or is 1 or 2 hours enough? Because what is the effect size of any chronopharmacology of what you will be doing when you kind of stratify maybe your cohort according to chronotype? So what if we are 1 hour off? Is it really making a big difference in any treatment or any diagnosis? This is one point. And the second point is maybe a question for the bioinformatics people. So what do we have anything, because we are doing a lot of blood sampling now from different kinds of patients, from shift workers and so on, and sometimes, and you can imagine if people have no rhythm at all, and we come to the amplitude issue, then the phase which is predicted by Zeitzeig or Lasso is somehow arbitrary. But when you get only one value, how can we know whether we can trust this value? Do you guys have any measure for us to say, yes, this is a reliable phase? This
John Hognes has a comment. You don't need to know how to say quite super accurate phase, and the one or 2 hours would be enough. Okay, so I also completely agree.
But I want to comment on this because I think it is a very important question.
Yeah.
See, if we say, well, 1 hour, what is 1 hour? We have many discussions about standard time versus daylight savings time. Well, that difference is 1 hour.
Okay.
And in that area, we believe that 1 hour difference in phase is very important. For chronotherapies like light therapy or melatonin therapy, maybe you only need 2 hours. And actually, I think that's probably the case. But it's not an easy question to really answer. And same for, can you fall asleep or not? If people try to sleep in the middle of the wake maintenance zone, they may have a very long sleep latency.
Yeah.
If they go to sleep 1 hour later, it may be fine. So I don't think we can simply say 2 hours is enough or 3 hours is good enough. I think that's too simple.
Yeah, so Derk Jan's comment is related to some kind of application. We can, evaluate the effect of the, times, summertime shifting, time zone shifting. And also, this accuracy discussion is related to the second point of discussion, like phase detection. How should...
Can I have one comment?
Yeah, Takeshi, please.
I'm not familiar with the human case, but it is possible to put the genome information to evaluate the genetic variation in human to combine with the transcriptome data?
Probably it's possible, although, scale is very limited. Yeah. But it's totally
have
possible.
Enough information on the circadian clock gene in human genomes.
Right. So currently, GWAS study has been done by using the question-based, phenotyping, like a morning person or a night person. But it's totally possible with this kind of information. But the detection is limited. So therefore,
But I guess that we have to distinguish that the physiological effect by the entrainment and the genetic variation in human beings. If not, it's very difficult to accurate estimations.
Okay. Right. So, so could we a little bit, so Achim, do you have a comments?
No.
Okay, so we have we have discussion about the phase, and also some discussion about the target, which is brought by Derk Jan. But do you have any comment on the amplitude? Because amplitude is quite missed in our field because it's a little bit difficult to detect, and then we a little bit difficult to distinguish the markers or real amplitude, or output of the circadian clock and the real amplitude. So therefore, do you have any comment?
Well, Jake, for example, has done some interesting work on cancer cells and looking at rhythmicity in a multivariate way, and came up with measures which he didn't. And if I misrepresent your work, Jake, I'm sure you will correct me. And he didn't really use the word amplitude, but maybe a better word, like, how much rhythmicity is there in that multivariate space. And so I think we may come to measures that better describe the overall amplitude of those rhythmic features. And that may give us better information about the state or the circadian state of that target. So I think it's time to work more on that. But maybe Jake wants to say something about that.
Yeah, Jake, do you have comments?
Yeah, I mean, I guess I would say, you know, all these methods, all the sort of organization of these data sets are designed to predict phase. They make an assumption that a phase exists and then predict a phase. They're not, you know, we don't necessarily, in a sense, have a ground truth in the same way about amplitude. And so I think it's harder. We're kind of coming at it from the side in a way. The other thing, though, about what Achim said was, you know, from a single sample, it's really hard to say and get an amplitude. If you have in your head the plot I showed earlier, which you probably don't, but you just imagine a circle with a point at the center, you know, is that a lack of amplitude, or is that the edge of a circle that's just displaced, right? So how do you know from one sample what that is? So, you know, even aside from the question about like, why do we want amplitude? What is the evidence that amplitude is going to be important for this application? That there's technical difficulties, I think, that we haven't addressed yet.
I think we want to have amplitude. I think amplitude is maybe even more important than phase, in my opinion, because we have so many problems with elderly people, with people in the ICU. I mean, if we measure, for example, around the clock in the ICU, the clock means that they're completely flat, these people. And people, I mean, John Hoganish and others are trying to kind of equip the ICU to kind of stabilize the rhythms. We want to follow that progress. But I think but one problem to develop something like a 1 or 2 sample method for measuring amplitude is that we don't have a gold standard for amplitude right now. So we cannot train against something. So when you want to do supervised learning, you need to have an outcome variable, but this we don't have for amplitude, or at least not. Maybe Derk Jan knows. I talked to Chuck Seisler and he said we don't have that. Maybe, I don't know.
Well, if Chuck Seisler says that we don't have it, what can you say?
Okay, yeah. You're right. You know, so John have a comment, you know, body temperature might be the good candidate for the amplitude measurement, but, you know, body temperature may differ among the person to person. So therefore, you know, amplitude information is some kind of missing in the field. Or maybe, you know, sleep can be, you know, staged every 1 minute or every 30 seconds. So maybe that kind of information can be used to normalize the amplitude information. So, but you know,
those data sets need to exist, right? I think the issue certainly that we've run into, and I think the same that Jake was saying before, is that in the existing circadian transcriptomics data sets that we're using to develop and train these algorithms, there isn't that amplitude metadata. So that needs to exist in order for supervised learning to take place.
Okay. So, and also recently, which is related to number 4, recently, you know, long-term recording is possible by using the Apple Watch or Fitbit. So which give us about some kind of a amplitude change over the time in individuals. So maybe you can, use such kind of information, kind of a relative amplitude information to this. So, Derk Jan, do you have a comment on the discussion number 4?
Yeah, well, I think many of the issues we discussed with respect to the one sample approach are relevant to the long-term approaches as well. Of course, there are now many exciting opportunities to do long-time data acquisition using apps and clever devices, and that's all wonderful. But those data are also subject to masking effects from sleep, activity, light, food intake, and we need to take those into account. And likewise, we need to know what is it that we actually want to measure. In many cases, of course, these data are collected under what we would consider diurnal conditions. And the data we get can be considered just a reflection of diurnal rhythms and not necessarily of endogenous circadian rhythms. And of course, there is a long history in people trying to develop methods to demask those kind of long time series data sets. And I think these same problems are still there also with our new devices. But I'm also quite optimistic that one can develop methods that would allow you to extract meaningful information from those long-term time series.
Okay. So, so maybe Achim, you have a comment, but because of the time limitation, I'd like to bring some audience to give a question to us. So, Catherine, could you pick up the some question from 50 question which is posted in the Q&A? I don't have a time to look at all of them, but Catherine, do you have a pick up some how to say audience?
Yes,
Bring them
I
mic.
will select Thomas right now. Your microphone is live. Thomas Kilka. Okay, it looks like his microphone will not be going. So next we're going to do Brian Altman. You can unmute.
Great. I really enjoyed the debate. So a question mostly to Achim, but to everybody. If we think monocytes are the most oscillatory population in the blood, can we use markers of monocytes from whole blood to rescale RNA-seq data to represent the number of monocytes in there to improve our sampling? I agree that we have a lot of whole blood samples out there that we'd like to sample and go back to stored samples. So it might be useful to take this approach. Do you think this might be possible?
Yeah, I think that's a good idea, but we would have to, I mean, it's only possible if the biomarkers would be basically monocyte specific biomarkers and not expressed in B cells and T cells. We may be able to revisit our monocyte transcriptomes and try to build a model using monocyte specific genes and then go back to the PBMC to assign times. But I think that as in our model, there's RefRB alpha and our RefRB beta and so on. This is also in the T cells and the B cells. So it could be done, but it's not easy.
Well, there are some cell deconvolution approaches that may actually allow you to get a little bit closer based on the whole blood transcriptome. So I think it's a good idea and a good question.
Yeah.
Catherine?
Okay, we have Hans Peter. You will be up in just a second, and your mic is live.
Hans Peter's concern is normalization. Can we use another time point to estimate changes, baselines of marker genes, sets of non-oscillating genes? Question to all. I'm just reading his comment.
Thank you, Achim. Can you hear me now?
Yeah.
Yes.
Sorry. So my more burning question is the amplitude issue. So let's assume we can measure everything. Then can we define what is the best measure of amplitude? Do we care about RefRB alpha or RefRB beta, absolute or relative amplitudes? Do we care about per cry? Do we care about cortisol? And this is, to me, also biologically not really clear. What is the amplitude of a circadian oscillator?
I mean,
so historically speaking, amplitude can be defined by the phase response curve. So, but it's a little bit difficult to measure. So,
to what? To light or to
light? Yeah, to light.
But again, also in the case of amplitude, it depends on what your target is. Right. if you want to have an assessment of the amplitude of the circadian pacemaker and relate this to its responsiveness to light, that would be your gold standard. It's a difficult experiment to do.
Right.
And of course, there have been measures of amplitude. You know, there is a paper, Megan Jewett is the first author in Nature several years ago. I think Chuck Seisler was the senior author, where several physiological variables were measured, including amplitude, core body temperature. And the claim was that these changes reflected reductions in amplitude of the central circadian pacemaker.
I see. Catherine, so I have a comment. So do we stop now or do we extend a little bit for Q&A?
I'm happy to extend for a few more minutes or a few more questions if you would
okay.
like.
Okay.
Presenters, are you okay with that? Okay,
yep.
Great.
Yep. So is there John Hogenish? Do you have a microphone?
Let me find him 1 second.
Yeah.
I don't see him on here.
Okay.
John, if you're available, will you raise your hand? Sometimes they log in with a different name, so
Did he?
I don't see him on here. Do you see another question until we find him?
Mm-hmm. Yeah, please pick up somebody.
Okay, let's do Dr. Clareman. I will get your mic. And you are allowed to talk now.
Chuck Seisler is here.
Hi. For amplitude, I think we have to consider that not all biological rhythms are sinusoid, and we have to consider how we're going to define amplitude for each of those metrics.
Right. Rosemary, Jake, do you have a comment?
I agree.
Yeah.
Okay, so yeah, you feel the difficulty. Okay, too. Okay. Rosemary, do you have a comment?
I mean, I think that once the outcome is well defined, it becomes at the risk of making it sound overly simple, a straightforward problem to develop an algorithm to try to predict the outcome measure of interest. I think the tricky thing here, especially when we're talking about amplitude, is defining what we mean by it, defining in what way is it biologically relevant, right? So we could measure amplitudes of all sorts of things, right? Just simply defined as peak to trough, but it needs to have some biological outcome that we're interested in, right? Some ramification. So I think those are the tricky things that need to be addressed.
Okay. Any other questions? So Catherine, could you pick up some?
Yes, I have. Charles, you are allowed to ask your question. Maybe, there we go. Charles, you can go ahead and ask your question.
Yes, can you hear me?
Yep.
Yep. Yes, I wanted to comment.
So my question was directed towards Derk Jan, who elegantly showed over the years the role of sleep in reducing what otherwise would be identified as circadian or at least diurnal changes in gene expression. I wondered about subtracting out sedentariness as opposed to sleep per se. Whether I saw you had constant routine on the slide. So what's the role of inactivity as opposed to sleep?
Yeah, no, I think that's a very good point. And as a sleep researcher, I always talk about the sleep-wake cycle. But the sleep-wake cycle is, of course, much more than just the sleep-wake cycle. It's also a cycle of being in the upright and the supine posture. It's also a cycle of feeding and fasting. And in the kind of experiments we have done, we haven't really separated out the separate contributions of all of those factors. But of course, it is true that in real life applications, most things shift with the sleep-wake cycle. So that includes posture and that includes feeding and fasting. But from a physiological perspective, you're absolutely right. And we don't, I mean, Kleitman showed back in the 1930s that a good way to lower your body temperature is just by lying down. You don't have to fall asleep to lower your body temperature. So it's clear that it's not just vigilant state, but there are other factors involved as well.
Thank you, Dr. Jan. So Chuck Seisler is here, right? So do you have some question or comments?
I believe he just asked his, was it Charles?
I think.
That was Tom Kilduff, I thought, who just asked.
Yeah, that was Tom.
That was Tom, yeah.
Yeah, Chuck is here.
Okay, who would you like to ask the question? I want to make sure they're unmuted.
Seisler? Maybe not.
Well, he put some comments on the chat, actually.
Yeah.
So.
Okay. Okay. Maybe difficult. Yeah, Zoom is a little bit difficult to have a question. Okay. So, all right. In the end of this discussion, I just want to have the comment of small comment from each speakers, I mean panelists. So starting from Takeshi Izawa, reverse orders. Okay.
Okay. So my comment is just that I'm looking for that collaborator in this field, especially application using the field transcript form data. Please send me some email if you are interested in. Thank you.
Okay. And then Rosemary.
Oh, no, my thoughts are very similar. I love collaborators. And, you know, as a computational researcher, I'm always looking for experimentalists to work closely with. So I would echo the sentiment of looking for collaborations. I think that this is an exciting field. I think that we're now at the point where we can collect incredibly rich data to drive forward methods development. So I think that we're only going to see increases in accuracy and more interesting applications as the field progresses.
Yeah. So, Achim?
Yeah, I want to use my time to thank you, Hero, and the other panelists for preparing an outstanding session, I think. And my scientific comment is, I think we should really work hard on the amplitude project. And maybe my 2 pence would be, maybe the gold standard of amplitude will be molecular amplitude, like gene expression, and not core body temperature or anything physiological, because it seems to be, at least what we see in the constant routine upon sleep deprivation, that the rhythms of clock genes are almost unperturbed by at least sleep deprivation. Let's see whether that is a possibility.
Okay, Jake.
Yeah, well, yeah, thanks, Hero, for letting me be a part of this. This has been a lot of fun. I guess the one point I might make is human health centric, but I think we need to do, you know, a better job of really establishing large scale evidence for chronotherapy. So Achim kind of alluded to it. How accurate does it really need to be? And, you know, can we, you know, simple is always better than complex, and universal is better than specific. So where are the cases where we really need, you know, within an hour, and what are the cases where we can say, you know, just take it before bed, it's good enough.
Okay, so finally, Derk Jan.
Yeah, I just would like to say that I think this session is evidence that this is a very good format to not only get clarity about, you know, where we agree, but also clarity about areas where we need to build consensus and get more agreement. And actually, it's quite remarkable to me how much we all agree. And if we read our papers and write our papers, that often is not the best way to have these kind of discussions. And I think this format has proven to be an excellent way to move the field forward.
Exactly.
Okay, so so I'd like to thank all panelists and also all audience who put the question, which is not answered yet, and also who give some have some patience to hear all of the entire discussions. Okay, so today we discuss about some topic, but some topics is still missing. For example, standardization, which is important to extend this field to the society, to the entire society. And also, we have not yet discussed about the non-invasive method to measure one-time point-based measurement by using non-invasive method. And also, we are missing some applications, not only human, but also to the other field, to other organisms. So we think as a community, we like to, closely collaborate with two others, and also agree on some consensus. And then I hope this kind of a diversity can bring or create some important application based on the single time point measurement. So thank you all.